Assalamualaikum and hi everyone. So today we are going to enter a new topic which is mutation. So in this topic, there will be 2-hour lectures and 6-hour tutorials. The learning outcomes for this topic are as follows. 7.1 Mutation Classification and Types. At the end of this lesson, you should be able to A. Define mutation, B. Classify mutation to 1. Gene or point mutation, 2. Chromosomal mutation, C. State 2 types of mutation, 1. Spontaneous mutation, 2. Induced mutation. Example, exposure to mutagen. D. Define mutagen. E. State types of mutagen. 1. Physical. Example, UV rays and gamma rays. 2. Chemical. Example, colchicine and ethidium bromide. Mutation. What is mutation? When we mention about mutation, people often view mutation in a negative light. However, without mutation, people would not have their rich color vision and some would not have a tolerance for lactose. Beneficial mutation can be found throughout the natural world. As example, the ability to digest milk is because you possess a mutation in your DNA. However, some mutation can cause detrimental conditions such as Down syndrome and Klinefelter syndrome. The definition for mutation is a permanent change in the amount, arrangement, or structure of DNA of an organism. Mutation may lead to change in genotype and phenotype that is different from its parent. If you still remember, in the previous topic, genotype is the genetic code for an individual, and phenotype refer to the physical appearance of an organism. Mutation can be inherited, and it can also create genetic diversity. If the mutation occurs in gamete cells or reproductive cells, they will be passed from generation to generation. Occurs in somatic cells. Somatic cells refer to all cells in the body except reproductive cells. So if mutation occurs in somatic cells, they are passed on to daughter cells produced by mitosis. As I mentioned earlier, not all mutations are harmful. Some mutation can be beneficial. As example, is the ability to digest milk. So lactose is the sugar found in milk. Lactose tolerance and lactose intolerance. About 75% of the adults worldwide are lactose intolerant, means they cannot digest the lactose found in milk. Those adults that can tolerate lactose can do so because of a genetic mutation. Individuals who have inherited the mutated gene for lactose tolerance continue to produce the enzyme that break down lactose. In conclusion, mutation in human genome allow many adults to digest lactose and drink milk. So in conclusion, not all mutations are harmful. So this is the symptoms for individual who having lactose intolerance. 7.1b Classify mutation to gene or point mutation and chromosomal mutation. So, mutation can be classified into two, gene or point mutation and also chromosomal mutation. Under gene mutation, there are base substitution, base deletion, base insertion, and base inversion. And under chromosomal mutation, there are change in number, or we call chromosomal number alteration, and also change in structure, or we call chromosomal aberration. So under the change in number, you will see aneuploidy and polyploidy, while under the change in structure, there are translocation, deletion, inversion, and duplication. So in this hour, we will be focusing more on gene or point mutation. And for chromosomal mutation, you will learn in the next hour of lecture. 7.1c state two types of mutation, spontaneous mutation and induced mutation. There are two types of mutation, which are spontaneous mutation and induced mutation. Spontaneous mutation occurs because of mistakes in base pairing or arrangement during DNA replication, or because of non-disjunction, while induced mutation can be because of the external factors such as high temperature and high light intensity, and also because of mutagen. Mutagen can be chemical or physical. The example for chemical mutagen are mustard gas, colchicine, and ethidium bromide. For your information, colchicine is very dangerous. Colchicine can prevent the formation of the spindle fiber in the nucleus during cell division. And the example for physical mutagen are ultraviolet, X-ray, alpha ray, beta ray, and gamma ray. Two types of mutation, spontaneous mutation and induced mutation. Spontaneous mutation is because of the mistakes that happen spontaneously during DNA replication. This mutation occurs naturally, about 1 in every million to 1 in every billion division. And it is due to low level of natural mutagens. Meanwhile, induced mutation is caused by exposure to mutagen, and these substances cause a much higher rate of mutation. So what is mutagen? Mutagen is a chemical or physical agent that induces mutation, and this substance can cause damage in DNA or alters DNA bases. As I mentioned earlier, mutagen can be divided into two, which are chemical and physical mutagen. And you must know the example for each of the following types of mutagens. So, the example for chemical mutagens are colchicine, ethidium bromide, and mustard gas. While the example for chemical mutagens can be either non-ionizing rays such as ultraviolet ray, and also ionizing rays such as X-ray, alpha ray, and beta ray. Next, learning outcomes for 7.2 gene mutation. At the end of this lecture, you will need to be able to A. Define gene mutation, B. State the 4 types of gene mutation, C. Explain 4 types of gene mutation. 1. Base substitution, 2. Base insertion, 3. Base deletion, and 4. Base inversion. D. Explain base substitution. Example, sickle cell anemia as missense mutation. E. State the effect of base substitution, missense, nonsense, and silent mutation, and base insertion and base deletion, frameshift mutation. F. Explain base insertion and base deletion as frameshift mutation. gene or point mutation. Gene or point mutation refer to the change in nucleotide base sequence of DNA molecules that represent a gene. So gene mutation is a change in nucleotide base sequence, in nucleotide base sequence of DNA molecules that represent a gene. So example of nucleotide base are guanine, adenine, thymine, and cytosine. Gene or point mutation occurs when it involves only a single base or single nucleotide pair of gene. This will lead to the change in amino acid sequences and thus may change the protein or enzyme that is being produced. The modified or different protein produced as the effect of mutation may not function as normal. So in this slide, you must remember the definition for gene mutation. So a change in nucleotide base sequence of DNA molecules. Four types of gene mutation. So under gene mutation, there are four types of mutation, which are base substitution, base insertion, base deletion, and also base inversion. So this is the animation for gene mutation. So over here we have base insertion. So we insert a base. Over here we have base deletion. So we delete a base. And over here we have base substitution. So we substitute a base. So you must remember and you must write down base in the front. Base substitution, base insertion, base deletion, and also base inversion. Next. Explain four types of gene mutation. Base substitution, replacement of one or a few base in DNA sequence by another base. So the keyword for base substitution is replacement. So we replace one or a few base with another base. Base insertion. So the keyword is insertion or addition of one or a few base in DNA sequence. Base insertion, we insert or we add one or few base. Base deletion, removal of one or a few base in DNA sequence. And lastly, base inversion. Base inversion is reversal of two or more base in DNA sequence. So for each types of gene mutation, you must remember the definition. So in this slide, there are three effects of base substitution, which are A. Missense mutation, B. Nonsense mutation, and C. Silence mutation. The effect of base substitution are missense, nonsense, and silent mutation. So what is missense mutation? Missense mutation results in the change of one amino acid with another amino acid during protein synthesis. Okay, so I repeat, missense mutation is the change of one amino acid with another amino acid during protein synthesis. So we will look at this. So wild type is the normal sequence of mRNA and also protein being produced by translation. And over here is the sequence that is having mutation. Okay, as examples, the wild types, the sequence of mRNA is GGC. So, if we refer to the amino acid table, GGC code for glycine. But over here, the mutated sequence, you have AGC. So, suppose you have G, but instead, G is being substituted with A. So now, the new sequence or the new codon is AGC. So if we refer to the amino acid table, AGC refer to serine. So missense mutations results in the change of one amino acid with another amino acid. As a result, the activity of an enzyme or hormone might decrease or be destroyed. So the example of missense mutation is sickle cell anemia. So for this one, you will learn more in details during tutorial class. The next effect of base substitution is nonsense mutation. So as its name, nonsense mutation changes a codon for an amino acid into a stop codon. Stop codon. So if you still remember, in topic 6, there are 3 stop codon, which are UA, 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 U, A, UA, and UA, UGA. So this is stop codon. So stop codon will cause translation to be terminated. So nonsense mutation changes a codon for an amino acid into a stop codon. This will cause translation will be terminated prematurely. As a result, it will produce short polypeptide and lead to non-functional protein. So we will see the example. So this is the normal sequence. So we have AAG over here. But if nonsense mutation occurs, you have UAG. So instead of A over here, A is being substituted with U. So now you have UAG. UAG code for stop codon. So the translation will be terminated prematurely. As a result, you only get methionine. But over here, you have AAG. AAG code for lysine. So this is nonsense mutation. Changes a codon for an amino acid into a stop codon. And the last effect of base substitution is silent mutation. Silent mutation refer to the codon is changed. The codon is changed, but it has no effect on amino acid sequence because it code for the same amino acid. Okay, so silent mutation, although the codon is changed, but it has no effect on amino acid sequence because the same amino acid is being produced. So as example over here, we have GGC. So GGC code for glycine, and over here we have GGU. GGU also code for glycine. So instead of having C over here, but you have U over here. So C is being substituted with U. But although the codon is being changed, but it's still code for the same amino acid. So this is why we call silence mutation. And lastly, we will see the effect of base insertion and base deletion, which is frame shift mutation. So, base insertion and base deletion can cause frame shift mutation. So, what is frame shift mutation? Frame shift mutation will occur if the number of nucleotide inserted or deleted is not in the multiple of 3. So, in other words, frame shift mutation will occur if we insert or we delete nucleotide 1 or 2. If we insert or we delete nucleotide 1 or 2. Come again. So, frame shift mutation will occur if we add or we delete 1 or 2 nucleotide. But if we insert or we delete 3 nucleotide, frame shift mutation will not occur. So, if the number of nucleotide inserted or deleted is not in the multiple of 3, it will resulting the triplets or the codons are wrongly read from the point of mutation and differ from that of normal gene. Frame shift mutation has serious effect because it causes all amino acid at and after the point of mutation to be changed, and thus producing different proteins. So I would like to repeat over here. If we insert or if we delete 1 or 2 nucleotide, frame shift mutation will occur. But if we insert or we delete 3 nucleotide, frame shift mutation will not occur. And you will learn further about frame shift mutation in the tutorial class. I think that's all for today. I hope you understand, and that's all from me. Thank you.